following question. Now, what I want to do is I want to just run through the 30 odd years that we've been working in the Carron, just to give you a flavour of what we've actually been going through, the problems we've faced and how we've dealt with them. And then that will lead on to the work that Victoria has done looking at DNA samples from the various fish in the river. I'm sure a lot of you will know the sort of history of the Carron. It was an excellent sea trout river in particular, and was also quite a good salmon river. But in the 1990s, the stock pretty much collapsed. Very quickly, it wasn't a slow process, it collapsed very quickly. And it went down to as low as no salmon and 5 grills in 2001, so at the end of the 1990s. And I can assure you, at the time, I was trying hard to catch fish, and I was struggling to find any. So there was certainly a big collapse in the stock. The condition of the river, it was a case of trying to find out what the problems might be. We looked at the general habitat in the river, and there was nothing wrong with the habitat. The habitat was perfect. There was plenty of good habitat for juveniles, for fry and parr, but the problem was, as electrofishing showed, there were hardly any fish in it. We electrofished lots of sites over the period through the 90s and into the 2000s, and it was a struggle to find any fry or parr. So that was the real issue we had, was not that we had habitat issues at all, it was the fact that we just didn't have young fish in the system. And of course, if you don't have young fish, you're not going to have smolts produced, and therefore you're not going to have fish coming back. It's quite simple to consider that. The reasons for the collapse, lots of things tend to be blamed for salmon problems. We certainly felt at the time that the biggest problem we had was red washout, major spates in the winter washing the eggs out. It's a very mobile river, the Carron, a lot of gravel. The main spawning areas are in the lower part of the river, from Loch down to the sea. That's the most gravelly part of the river. That's undoubtedly where a lot of the spawning takes place. To try and put a sort of handle on the issues around red washout, there was a piece of work done by Dr. James Butler, who was the scientist, the biologist of the West Ross Fisheries Trust back in the early days. He was concerned about red washout, and he set up a field trial where, for a lot of the rivers around Western Ross, he planted out beads in the bed of the river, tethered to a stake. Beads of about the same size as a salmon egg, buried to the depth that a salmon would lay its eggs. And then he went back after the winter to see how many of these beads had been washed out. If they'd been washed out, they'd have been hanging on the thread. 5 sites in the Carron were looked at as part of this trial, and he found red washout in many of the sites around Western Ross. And for the 5 sites in the Carron, all 5 sites were washed out. And that was various different parts. Some were spawning barns, some were main stem of the river. Now, the maximum flow that year, we have a SEPA gauging station on the Carron that's been in operation since 1979. So it's possible to get flow rates every 15 minutes over that period. So I looked at the data from the gauging station, and in that year that James Butler did his work, the maximum flow was 120 cubic meters per second. And that happened in, I think it was February. So that would have been a time when certainly eggs or alevins would have been in the gravel, probably eggs at that time. In the 5 years before we had this collapse, we had 5 years of major spates. The first one in 1988-89, the maximum flow that year on a day was 330 cubic meters per second. So if you compare that with 120, it's a very much bigger spate. Now that in itself, if it had been 1 year, that would have been a major problem, because you've still got bigger fish in the river, you've got fish at sea to come back. But we had 5 years of big spates. The next 4 years had pretty major spates as well. Not quite as big, but big enough to cause damage. So what I reckon happened was that over that period of time, we'd simply lost what fish we had in the river, and because nothing was going out, we'd lost any fish coming back in to replenish. Now that in itself, if that 5 years was all the spates that we would have, then the river could certainly have recovered. But from the SEPA gauging station right through the whole of the 90s, there wasn't a single year when we didn't suffer a big spate in the winter. So the chances are we were getting this continual washout. Now that has continued right through until now. Between 1979 and 2012, we only had 4 days when there wasn't a big enough spate to wash eggs out. In the last 4 years, we've had big enough spates to cause a lot of gravel movement, and I go down to the sea pools every year in the summer to fish for sea trout and finnick, and I notice the differences in the amount of gravel, and I reckon between 2,000 and 3,000 tonnes of gravel has come down over the last 4 winters. And of course that's coming from areas where fish have spawned. So we reckon that was the real problem, and it was a case then of what do we do about it? What can we do to try and restore the system? And we were trying to simply get some fish back. We weren't looking to create huge runs of salmon. We simply wanted to get a few fish back, so at least a few anglers might have a successful day. So stocking was an obvious potential way to go, but the issue we had was that it was going to be very difficult to get any broodstock out of the river because the stocks were so low. We started stocking in 1995, and between 1995 and 1999 we were only able to put a maximum of 10,000 fry into the river, and that was from the very small number of eggs we could gather from wild broodstock. It became very evident very quickly that that was never going to be enough to make a difference. We didn't see catches improving. There might have been some extra fish back in the river, but it wasn't reflected in catches. So we had to think of a way of generating a much bigger production of eggs that would allow us to perhaps get some more fish coming back. And with that in mind, we went down the captive broodstock route. So we kept some eggs from the few fish we could catch, and we reared them right through in fresh water, incubated them, hatched them out, took them through right through to maturity without ever going to sea. And it certainly worked. We were able to generate reasonable numbers of eggs. So the first major stocking, the first year that these captive broodstock came on stream was 2001. So instead of having only up to 10,000 fry to go in 2001 we had 159,000 that we could put in, and that had come from the captive broodstock. Following those fish through to go to sea as 2-year-old smolts, which the majority of them would have done, then coming back as grills, it would have been 2004. In 2004, a rod catch leapt up to 141. So there was a major increase in the rod catch. Over the next few years, as the captive broodstock produced more and more eggs, then we were able to stock more and more fish, and the catches went up accordingly. There was a very close correlation between the number of fish we were stocking in those early days and the number of fish we subsequently got in the rod catch. So that seemed to be working. The first year that we would have actually got, and as these catches went up in 2007, we actually had the record catch at the time of 261 fish. That was the most fish ever recorded since 1952 caught in the Carron. In 2008, we would then have started getting fish that were a combination of wild spawn fish, because of these extra fish that we'd had coming in 2004, and continuing with the captive broodstock. So from then on, it would have been much more difficult to distinguish between what was coming from captive broodstock and what was coming from natural spawning. Talking about catches, in 2010 and 2012, we caught over 400 salmon in each of these 2 seasons. And that was totally unheard of in the Carron. We'd never have had catches of that sort of size. Those were good fishing years, to be fair. I think most rivers around Scotland did well in those 2 years. But you're only going to do well if you're generating a big enough smolt run. So that seemed to be going pretty well. This particular captive broodstock went on until 2015, and then we stopped it at that stage. We also developed a second captive broodstock. This time it was a more natural cycle that the fish went through, and this was in collaboration with the local salmon farm. They were keen to look at working with Scottish stock. They asked us if we could provide them with some multi-sea winter eggs, and they would give us eggs back. So if they took this stock through, then we'd be able to get some eggs back. So it seemed to be a good way to go. I provided them with batches of eggs over a 4-year period, and these were taken from several pairs of multi-sea winter broodstock. These eggs were taken out to North Uist. They were hatched and brought through in freshwater tanks in North Uist, and then transferred into sea cages to be brought through to maturity. and I was then getting stock back from these fish. The fish did very well. The egg quality that came back was good, and the fry we generated from them were good fry. This particular broodstock went on for 4 years, and it was discontinued in 2018, partly because policies were in place that meant you had to use wild fish taken from the river. So it meant that we had to discontinue that particular broodstock. Now, what's going to be important in all of this, and I've been emphasising this in recent times, is that rather than concentrate on angling and getting fish back for anglers to catch, which certainly anyone who's a keen angler would like that to be happening, but we've got the stage that conservation is more important. It's more important to get fish back to replenish stocks or try and build stocks back up again, because a lot of our small rivers, particularly ones on the west coast, are certainly in danger of extinction. We're certainly in very great danger of some of the smaller rivers losing their stock entirely. So with that in mind, what was going to be important was that the fish we were bringing back, or getting back from the breeding program, had to then give rise to healthy offspring. They had to give rise to viable offspring, otherwise you're not going to be able to conserve the stock. It simply wouldn't work. What I've done over the years is we did some tagging work back in 2006, 7 and 8, where we tagged 105,000 autumn fry at an average weight of 4 grams. The 2008 fish, which was 40,000, I was able to monitor them going through a screw trap. We run a screw trap at the bottom end of the river and have been for the last 18 years or so. Because these fish were fin clipped, I was able to identify them going through the trap as being these stock fish. What I found was that of that 40,000 fry that we tagged, 137 smolts went through as 1-year-old smolts. They were very small smolts, smaller than you'd naturally expect to get. 822 went through the trap as S2s, and they were identical in every way to all the other smolts, apart from the fact that they had a fin clip. So that's how we could identify. And then only 13 went out in 2011, which were S3s. So they were 3-year-old smolts. So the bulk of these stocked fish were going out as 2-year-old smolts. Now that is slightly different from what you would naturally expect to get in the river, where you get a mix of 2-year-old and 3-year-old. That because these fish were reared, they're obviously growing faster than a wild fish would. So they're getting to the trigger size to become a smolt a bit earlier. So we're getting these fish out. Now, we tested the efficiency of the trap in 2011 and 2013 by releasing stocked smolts that I could recognise and monitoring the number that went through the trap. And it worked out almost exactly the same in both years, at between 16 and 17% of the fish that were released from the release pond went through the trap. So applying those figures to this fish that we monitored in 2009 and 2010, meant that over 5,000 smolts would have left the river from that stocking in the autumn, which is about 14.2%, I think it worked out at. So these fish were doing well. So we knew that the freshwater part was working well. We knew that we were getting the fish going out, and obviously some of these fish then come back as clipped fish in the following years. But that system of monitoring, and monitoring is very important with any stocking program, you must try and work out exactly what's happening. It's all very well, in the early days we were pretty sure that there was a very, very strong link between the stocking and the fish coming back. But scientifically that's not strong enough evidence. You really need to be able to prove what your stocked fish are doing. So the next step was to go down the route of DNA sampling, taking clips off the fish, and then with the help of Victoria and her team to actually look at the DNA to match up, to see if we could match up the stocked fish with what was going back. So in 2011 we started keeping clips from all the broodstock that we used. So every pair that I stripped, I would take a clip off the tail of the cock and the hen, and that would all be recorded, it would all be saved to be looked at at a later date. We also then took clips of smolts leaving the river via the screw trap, and that was a total of 200 each year from 2013 onwards. And then we also took clips of returning adults. in the rod cap. And that was a case of making sure that anglers knew to take a little clip off the fish that we could retain, and then that could be looked at. So that means that we can then look to see whether there's a match between the stocked fish and the fish coming back. Now, so that leads us on very nicely to the DNA part of the study, which is the main reason for coming tonight, is to go through the results that Victoria has put together to look at a very strong scientific method of determining how well the stocking is doing. If DNA is good enough to convict criminals, then it's good enough to sort out what the fish are doing. So I'll is that still with me?
Yeah, keep going, Bob.
So I'll tell you, this is very unusual to do it for such a short time. But obviously you can ask me questions after this, and we can look at a few other things perhaps. So it's been a long haul. It's very time consuming for any of you here that's ever reared fish. You can never take your eyes off them, because if the water stops, the fish die. If anything goes wrong, the fish die. So you really never get away. I think the last time I had a holiday was 1996, much to my wife's annoyance. I have a family that have grown up accepting the fact that my fish come first, and they've all been very good about it. Many they've all disappeared; they're all away at different places. I don't know if that's got something to do with it. But it does take a lot of effort. It's very time consuming. It really is a labour of love, and as far as I'm concerned, I've got such a passion for salmon and catching salmon. Not killing salmon anymore, but certainly catching salmon. That it is worthwhile doing, and I would like to think that this is a tool in the toolbox that should not be dismissed, because there's going to be situations where it might be the only way forward for some rivers, particularly those where the stocks have got down to very low levels. The danger with a lot of this now is that if stocks are allowed to get too low, this whole process becomes more difficult. If you're relying on taking fish out of the river to use as broodstock, and as I said, you need the numbers of eggs, it's a numbers game. If you're dealing with small numbers, you're not going to get the benefits because of all the pressures that we have on fish. And we have huge pressures on salmon just now. I've mentioned the winter spates that we get. We also have a very large seal population just beyond the mouth of the Lochs Institute. My neighbour counted 340 hauled out 2 summers ago. And if we then get dry weather in the summer, which, believe it or not, in the west we actually do, we do get some very dry spells in the summer. The river goes very low and the fish don't come in, and obviously they can be gobbled up by these seals. So there's a lot of pressure on fish, and we need to, because of that, we need to act sooner rather than later in terms of saving particular populations. And it's more of an issue with, as I say, our smaller rivers, particularly those on the west coast, than maybe the bigger rivers that still have a reasonable population of fish. So I'll pass you on to Victoria now, who will then go on to, you'll find that as interesting as I do.
Bob, thanks very much indeed. Your work is heroic. While Victoria's sorting out her computer, I'll make a quick introduction. Dr. Victoria Pritchard at the IBFC at UHI in Inverness. She's an evolutionary biologist, looks at the genomic, morphological and behavioural divergence in populations, the consequence of hybridisation, which is very important for the work she's been doing with Bob. Looking at her CV on the UHI website, it says that her work tries to answer the questions how the diversity of life arises. And I thought looking around the room tonight would be much food for thought for you here, Victoria.
I don't think there's that much diversity in this room.
And I can see about 3 or 4 diverse people that actually spot on. Do you need a microphone, Victoria?
Can everyone hear me, or is it best for me to have a microphone? Okay?
Take Pritchard for the microphone.
Okay. Thank you, everyone, for inviting me, and thank you especially to Bob, without whose incredible hard work we can do this research. And I suspect Bob has fridges and freezers still full of Carron salmon in his house. We have at least 2 fridges full of Carron salmon in our lab too, so there's an awful lot of material to be using. So I'm just going to give you a summary of the work that we've been doing this year, funded by, is it called the Wild Salmon Fund? Yes.
Wild Fisheries Fund.
Yeah, Wild Fisheries Fund, in collaboration with the River Carron Conservation Association. And what we're doing with this work is using fairly state-of-the-art genetic approaches to really understand how the stocking is maintained in the Carron population, but also what effects it might be having on the Carron population. And this work I'm doing at the Institute for Biodiversity and Freshwater Conservation, which is part of UHI Inverness. So before we start, I'm just going to give a really short discussion about stocking and genetics, because I think you all know that stocking is controversial, and one of the reasons it's controversial is because it can change the genetic composition of the salmon population that receives the stocked fish. One thing people worry about is if you take fish from the wild and rear them up in hatchery, they'll become genetically adapted to the hatchery, and then they won't be so adapted to the wild. But this is more of a problem when you're rearing broodstocks over multiple generations, which actually the Carron is not doing in this case. So, assuming that we have the blue wild fish on the left and the orange stocked fish on the right, another worry that people have that's been shown fairly regularly is that the stocked fish just don't survive and they don't come back. And in that case, you've wasted a lot of money doing your stocking program. There's already some evidence from tagging studies in the Carron that the stocked fish do come back. And I'm going to show you some additional evidence for that today. Another potential impact is that when you're introducing a large number of well-fed hatchery-reared juveniles into a wild population, they will be competing with the wild fry that are already in there. And if your rivers are at carrying capacity, there's going to be death because of this competition. And because your wild families will be many different sizes, sometimes you might get a whole wild family going, not surviving, and then you've lost the contribution of particular wild parents. But because generally your stocked families are bigger, you are less likely to lose the genetic contribution of the stocked fish. And I'm just going to introduce a term I'm going to use at the end of this talk, and it's a term that is effective number of breeders or effective population size. And to tell you what this is it's a measure of the genetic size of the population in effect, which is different from the actual number of fish in the river. So you can imagine a scenario where you've got a population of salmon in the river, but there's only 2 families. And then you compare that to a population in the salmon river, same number of salmon, but there's I think there's 10 families on the right. And it's quite easy to understand that the genetic size of the population with only 2 families is smaller than the genetic size of the population with 10 families. So just to try and explain that simply when I come to it. Okay, so we use the really fantastic archive of genetic material that Bob has been collecting from all the fish he stocked. all his broodstock, and it's from all the broodstock, all the returning adults, and many of the stocked fry and the stocked smolts in the river. So really amazing archive. And having this archive and combining it with genetics allowed us to address these four important questions. So the first question, really, what everyone wants to know is, do the stocked salmon that Bob puts into the river as juveniles, do they survive, do they come back as adults, and are they in the rod catch? And if so, what proportion are stocked fish coming back, and what proportion are fish that were spawned, fish that were born from wild spawners in the river? The third question is, what sort of genetic impact might the stocking have had? And Bob told you about the different broodstock strategies, so we're also able to look at whether different broodstock hatcheries have different effects. And this is just recapping the different broodstock that Bob told you about. The wild broodstock strategy, Bob takes females and males that are rod caught in the river, and then he takes them into the tanks, and then he crosses them when they're ready. For the Atterdale broodstock program, ran from 2012 to 2015, and in that case the female parents were kept in the hatchery, and they were used in multiple years, but the male parents were rod caught wild fish. And then he also talked about the newest broodstock, which were males and females, both kept in the hatchery, but in a more natural conditions, right? So they were ready to maturity in
the natural cycle of freshwater and seawater.
Yeah. Okay. So we had three different stocking strategies, and for most of these stocking strategies, Bob collected tissue samples from all the wild broodstock parents. There was just 2 years of the newest broodstock where we didn't have samples from parents, but we had samples from their known offspring. And then Bob also has provided us with genetic samples taken from all the adult fish that had been caught by the rods in the river between 2014 and 2021. So a really, really nice sampling of what was coming back to the river. I'm going to skip through this for anyone that's interested. This is our lab workflow. We take the fin clip, we extract DNA from it, and then we genotype it at 88 different genetic markers, which are across the whole Atlantic salmon genome. So then we've got basically a genetic fingerprint from each of our fish, and then we can do various different statistical analyses to address our different questions. The main thing we're doing is something called parentage-based genetic tagging, which is effectively paternity analysis for Atlantic salmon. So we take our adults that are returning to the river, and we take our genetic samples of the broodstock parents that could have produced those adults. So it's there's a range of years of broodstock that will produce the returning adults. And we ran a statistical parentage analysis that would use the genetic fingerprints and link any stocked fish that was returning as an adult. We could link it to its two broodstock parents. And then if we weren't able to link a fish to any of the broodstock parents, we inferred that that was a wild spawning, not a stocked fish. Obviously these are not salmon. Okay, so I said we had returning adults from 2012 to 2021, but I'm just going to focus on these 4 years between 2017 and 2020. And the reason for that is that we had samples from almost all of the broodstock that would have given rise to the adults that returned in those years. If you look at if you think about a range of smolt migration timings, the range of time the fish stay in the ocean, and when they come back, I'm confident that for these years we would have been able to discriminate almost all the stocked fish from all the wild spawn fish. So I'm focusing on those 4 years now. And the first nice result is, yes, the stocked fish do survive and they do come back to the Carron. And approximately half of the rod core adults each year were stocked by Bob originally. And the other half appear to have been produced by wild spawners in the river. So that's the first fear. Has not come true. The stocked fish do survive. We also compared the genetic diversity of the stocked fish and of the wild fish in each year, and there wasn't very much difference. Each year the stocked fish had a slightly lower genetic diversity than the wild fish, and in 2 years that was statistically significant. But it wasn't like a really large effect. So the stocked fish appear to be almost as genetically diverse as the wild fish. We also looked at this measure that I talked about earlier, the effective number of breeders, which is a measure of the genetic size of the population. So I apologise, the text at the bottom might be a little hard to read. But what I'm doing here is taking all the different possible combinations of stocked and wild fish and asking how many returners did they each contribute, how many possible parents are represented in those returners, and then what is the effective number of parents or the genetic size of the population. And for reference, the first column there is the wild spawning fish, the non-stocked fish, and then so I've put the orange line shows the level of each of those measures for the wild spawning fish, and then the question is the stocking, if you add the stocked fish to them, do you get an improvement? So the second column there is all the fish, so the wild fish and the stocked fish. And you can see, because half of the returning adults are stocked, you get double the number of fish come back. But when you look at the bottom figure, you're not getting a doubling of the genetic size of the population. So the stocking is contributing a lot of individuals, but it's not contributing so much genetic diversity. And then if you look at these next 3 points, these are the wild spawn fish with the Atterdale broodstock, the wild spawn fish with the Ewis broodstock, and the wild spawn fish with the wild broodstock. And you can see the different broodstock strategies have affected the genetic size of the population differently. So the Atterdale stocking strategy has actually decreased it a little bit, and this is because you've got a lot of females that are being used over multiple years, and so you've got big families from those females. Whereas the wild stocking strategy, where you're taking wild fish out of the river and crossing them, that actually seems pretty successful in increasing both the number of fish and also the genetic size of the population. So not only has Bob's information been able for us to show that the Carron stocking is working to bring back stocked adults, it's also showed us what might be the best broodstock strategy to use going forward for the genetic health of the population. So we were also able to use the genetic information to ask whether there are escaped Norwegian aquaculture fish, whether they are breeding with Carron fish, because we know that's a problem on the west coast of Scotland. And we do see, so here the orange colour is genetic material, genetic ancestry from aquaculture fish, and the blue is genetic ancestry from wild Scottish fish. And we do see that aquaculture fish are coming into the Carron and breeding with wild fish. And it's really quite hard in many cases. This is one of the problems with having aquaculture in the vicinity, is that you can't always tell the difference between aquaculture escapes and wild fish, and you definitely can't tell the difference between first generation offspring of escaped aquaculture fish and wild fish. This aquaculture material in the Carron is also, some of it is in the broodstocks, but the broodstocks are not contributing any more. That's just being contributed by the wild spawners. So this work has given us some nice new insights into how the Carron Restoration Stocking Programme is working. There are some things that this information that can't tell us yet. So we can see that half of the returning adults are stocked fish, but we don't know whether if the stocking wasn't happening, whether more non-stocked fish, wild spawn fish would be returning. That's something we can't ask with the data we have at the moment. We don't know whether the stocked fish survive and return at a better rate than the wild caught fish, or an equal rate, or whether they're actually surviving less well in the ocean. And the results can't tell us whether you need to continue stocking to maintain the Carron population, or whether now that it's secure enough that you could stop the stocking. And I'm sure that Bob has a lot of things he can discuss about this. So thank you very much for listening, and we have quite a big project team, but I just want to thank especially Dasha Svobodova, who has done most of the lab work for this project. Thanks.
Victoria, thanks very much indeed. That was super.
Sampling the smolts in the screw trap is that, as I mentioned, from 2013 we started collecting smolts from the trap, and that amounts to 200 every year. What I normally do is I take the first 20 smolts each day and take clips off them to be preserved for DNA analysis. So the 200 will be collected over a 10-day period. So it's not a case of just taking 200 smolts out on one day and sampling them. So it spreads them out over a longer period. Given the fact that that's 200 a year since 2013, it's quite a lot of smolts to get done. And the important thing about getting the smolts done, obviously a lot of people will be interested in how many fish come back. How many fish come back and are caught by anglers. And I think a lot of people who are interested in any form of stocking, that would certainly be in their mind, that they want the extra fish so that people can catch them. But as I mentioned earlier on, it's now more a matter of conservation. But it is quite important to try and ascertain where losses might be happening. And simply by looking at the returning adults in the rod catch, that doesn't necessarily tell you whether they are being lost at sea or whether they've actually never got to sea. They haven't simply left the river. So getting the smolts done will be quite important, because it'll give us a clue. We already had some indication from the tagging work, but as I said, that only included some of the fish, didn't include all the fish. So getting some DNA from the smolt run will give us an idea of how freshwater survival has been, how well that has been. And then we have the information from the returning adults, and we can then get an idea of where the biggest problem might be, whether it's before they go to sea or when they come back. What we certainly found from the tagging work, for the smolts going out was that a lot of these fish seem to have done perfectly well in fresh water. I mean, we had good numbers going out through the screw trap. So that gave us certainly an indication. We're getting more of the smolts done, will be good. We obviously have broodstock. I'm still gathering smolts, gathering clips every year. So every broodfish I strip, we take clips off, and I take clips off as many of the rod caught fish. We don't get all the anglers taking the clips, but we get as many as we can. So that's ongoing. So the number of samples that is still mounting. We're still getting more and more. And obviously, the more samples that we can look at, then the stronger the evidence becomes either way, whether the stocking is doing well or not doing well. What I should have mentioned earlier on, and I'll just throw this in at this stage, is that when I said that conservation is now more important than angling as far as I'm concerned, and I'm saying that as an avid, fanatical angler. I mean, I haven't deliberately killed a fish for a lot of years, and when I was a young man, I used to kill plenty, as everyone did. They fished to take fish home. It's important that these fish are doing well. Now, from the last DNA that Victoria did, there were 6 hens that appeared to be stockfish from the DNA. 2 of them came from the used broodstock, 2 came from the Attadale broodstock, and 2 came from the wild broodstock. I had caught these fish in the river when they returned, and as far as I was concerned, they were wild salmon. There was no indication from any of these fish that they were anything other than a wild salmon. But of course, the DNA showed that they were stockfish. So these fish were stripped, and I got 38,400 eggs, eyed eggs, from those 6 hens, at a success rate to the eyed stage of 99.3%. So as far as I'm concerned, that was excellent quality eggs. And I've been working with eggs for more years than I care to remember. And I know that a high percentage reaching eyed stage gives you a high quality fish all the way through. That's the indicator that you've got a good batch of eggs and a good batch of fish. So the survival of these fish was extremely high. Now, one of those fish was a 22-pound hen that one of our friends from across from Inverness caught. And on the same day, I actually caught a 20-pound cock in the adjacent pool. And I'd kept that hen, the eggs I got from that hen, separate from any others. In fact, I do that with all the batches of eggs. They're all kept separate until they reach the feeding stage, and then they're combined for feeding. So we can determine the genetics of each batch that goes out. But that particular fish produced 11,400 eyed eggs, and when it came to these fish reaching, or these eggs hatching, coming through and reaching the 2 to 3 gram size, I still had 11,200. So the survival was extremely high. Now, one of the arguments against stocking, which I can't quite understand, and there might be somebody here that could enlighten me, is when we're told that hens don't get a chance to choose their partner if you're doing it in the hatchery, because I would be doing it for them. I'd be matching a multi-segmented hen with a multi-segmented cock and a grilse with a grilse when it came to taking the eggs and fertilising them. I would like to know, if anyone can tell me, what is it that a hen salmon looks for in a cock when it's choosing a partner? What I liken it to is if you have a bitch in season that escapes, she'll mate with any and every dog she meets. And normally it won't be the same breed. So I don't understand how a hen actually chooses its mate. If anything, it's the cock that will choose the hen because he's going to chase away other fish. And he might be the strongest fish, but is it the size of the kite? Is it the size of the adipose fin? Is it his colour, his size? I don't think there's any choice going on there. When a hen is ready to spawn, and I know from many years of stripping eggs, once she ovulates and her eggs are freed into the body cavity, that fish has to spawn. If she doesn't spawn at that time, she will lose her eggs. And I know from I've had fish in tanks that have been ripe, and I've maybe been a day late in checking them, and there'll be lots of eggs at the bottom of the tank. Simple movement of the fish will shed the eggs without her making a red. So when she's ready to spawn, she'll want to spawn. She won't have time to decide on which male she's going to mate with. The other thing is that we get a lot of precocious parr fertilising eggs. I suspect that nearly every hen that puts eggs into a red will have precocious parr around fertilising eggs. Is she choosing to have precocious parr there? I suspect not. I suspect she's only interested in the bigger male who stimulates her to shed her eggs. So I don't buy into this business of choice. I would have to be convinced that there's actually something that the hen was deciding on. And would she refuse to mate with a cock that she didn't like the look of? Or when she was ready to shed her eggs, that was it. If there was a cock present to stimulate her, she would do it.
Yeah.
So that's just a little aside, and if anyone can enlighten me, I'd be delighted to hear.
I am not up to date enough with the literature to talk on this. I do know that it's pretty clear that there's quite often the hens have a lot of male parents of their offspring. Especially, I think we're definitely underestimating the impact of precocious parr. I think they're pretty important. There is some evidence, from what I remember, that actually the egg fluid that the females produce might favour some types of sperm over the others. So that's quite interesting. But I'm not up enough on the scientific literature to be able to talk with authority on Bob's suggestions here.
Okay, thanks. Can you stick your hand up if you've got a question for Victoria or Bob? Thank you. And could you say who you are and just a tiny bit of background so we know which direction you're coming from, please?
Okay, my name is Dr. Marion Bruce. I'm a molecular geneticist. I come from a family of fishermen. I'm now a director by Region in Tayside, which is working across the whole catchment to not just restore salmon to the rivers, but to restore the whole ecosystem. I'm just wondering whether you understand whether there's a real, well, what is the carrying capacity of the river, and also whether that's changed over the recent years with this increased water flow that's come through and as a result of the scouring of the gravel, if that has actually changed the carrying capacity of the river detrimentally.
I'll let Bob talk on this mainly, but I've been quite a hobby is in buying old salmon books, and certainly in the late 1800s, there was the rod catch on the Carron was very low, but it's not clear whether that was due to a coastal fishery or other things going on. But certainly the Carron rod catch has fluctuated a lot over at least over a century. But I'll let Bob give his opinion on this.
Yeah, I think undoubtedly it does change. As far as carrying capacity, we regularly electrofish, and we tend to do the same sites. We look at the same sites. And in 2000, we actually did a lot of sites in 2000, and we found very little in the way of young fish. What we tend to find when we electrofish is we pick up parr. We can pick up reasonable numbers of parr, but the fry numbers tend to be pretty low, and I suspect that's probably due to washout. The fact that any successful spawning that there's been hasn't been so successful in terms of the stock coming through. It is a difficult one to answer because we could maybe continue to increase the number of fish we put out to get to the point where you can't put any more in. I don't think it's an easy one to answer for anyone. And all I would go by is if you do electrofishing and you're finding very little, and you've got good habitat, which we have. We've got a lot of good habitat in the Carron, apart from the moving gravel that we get each winter. There's plenty of good fry and parr habitat. Then I suspect the river could carry a lot more in terms of young fry. But of course, what we're doing is, and I certainly favour putting fish in towards the end of the summer and the autumn, and I've been favouring that for a number of years, because the success rate of these fish, from what I can see, is good. Fish at that stage. fish going out at an early stage, and this would certainly be happening naturally as well, is that they're much more prone to predation. For one thing, we've got goosanders in the river. Any of you here that fishes salmon rivers will probably encounter goosanders. They are incredibly efficient goosanders. They're incredibly good mothers, and this is probably the biggest issue that we would have with goosanders. So they'll rear complete broods of a dozen young. If you start to think of a dozen young reaching fledging and maybe leaving your river in August, which ours tend to do, we've only got maybe 3 or 4 pairs in the river, and I see the damage that they do to smolts when I do the screw trap. So they're obviously around at the time when your smolts are running, but they would do an awful lot of damage when you've got small fry. These young goosanders, and I've watched them. I remember one year watching 2 broods, one on Sean's beat and one on the beat above. And they come down a pool in line and dive as they come down the pool. So they'll be mopping up anything that they can get hold of. And I would like, I don't know if anyone actually has a figure for how many young salmon it takes for a goosander to fledge. And I suspect it's a lot of young fish. So I suspect we're not at full capacity in the Carron. We're probably at capacity in terms of how many fish I want to rear and how many fish I want to put out. And when people ask, you know, what's the maximum number of fish I should put in, I suspect is how much can you afford would be the crucial thing rather than how many fish.
Okay, thanks, Bob. Sean, sorry, Victor. Sean, can you give a contribution
the
after?
question of ecology is very interesting, and it's something that Melanie Smith, Professor Melanie Smith, is very interested in.
Can you speak up, Sean, please?
Yeah, that Melanie Smith, Professor Melanie Smith, is very interested in. And Alice has been doing a huge amount of work planting at one end of the river. So in terms of nutrition and what the food is available, are probably as good as ever. On the forestry side, we're reversing the mistakes the Scottish Government made with forestry ploughing in the 60s, which has contributed hugely, we think, to the red washout. It's important to remember when we were at our lowest ebb that the fish farmers were getting the blame all the time, and actually what Bob has done in the Carron is create a smolt run to challenge the fish farmers to give safe passage. The other thing you should look at, and if you get in touch, we'll send it to you, there is a fantastic report from 1938 that Pascally did, the Scottish Government did, on sweep netting Loch Dule in February and in November. And the amount of fish that were in the system at that time, the old, you know, probably a cobble and that sort of thing, is quite remarkable. So our carrying capacity, in answer to ecology, is probably, even when Bob was at his peak of stocking, is still well below how many eggs would have been deposited in the river in 1938. And because so many eggs were deposited, many other species benefited along the line. And that's something that, you know, I would hope with the work that Victoria has done and Bob has done, that this will be forthcoming, maybe from the Scottish Government, to look at all the other projects that we've been doing on the Carron and all the other work. So yeah, we're very interested in that part of it. We're looking at the whole system and how going forward we can improve the nutrition, the feeding, and the forestry, and reducing, you know, these red water spades. And we have been ignored on this matter a wee bit, but I think that we've never been wrong about it. We didn't think that fish farming was the biggest problem. We challenged the fish farmers. We've had success in the smolt run, and we've had returning fish, and we continue to do that.
Yeah, that was my reason to actually ask it whether actually these kind of schemes are actually getting anywhere near the natural carrying capacity of 200 years ago within these kind of rivers before there was significant removal of fish at the mouth of the river or on line caught.
Yeah, can I come in here, because this is, do you want to come in? Or, this is really an important question because, as I said in my talk, you're taking, your captive fish come from really a relatively small number of parents, because you can't get, there's only a certain number of parents you can manage. But they're large numbers of well-fed fish, and you're putting them in the river on top of the wild fry. Now, if you're close to your carrying capacity, those fish are going to compete with the wild fry, and what you're going to end up with is fewer families in the river and less genetic diversity. So that is a real risk. Now the Carron, Bob and the others are pretty convinced that there's so much red washout that this in itself is limiting the population of juveniles well below their carrying capacity. But that's not necessarily the same in other rivers that might want to do stocking, and it is a big risk. So I'd caution about, you know, talking about how you know, putting in as many stocked juveniles as you can, because that's a risk on the genetic health of that population for sure.
So would it be generally kind of, you could generalise to say that if you were near to your carrying capacity, actually you would limit the number of stocked fish, but if you were really down at the very lowest end to keep the population to survive, then actually it could be particularly beneficial.
Yeah, I think you should be. This is something you should be considering. What would be a trigger for stocking, as well as being very careful to manage your stocking to maximise the diversity as much as you can. I'll let Colin come in because he's been wanting to talk.
Yeah, I'm Colin Gould from the Institute of Aquaculture in University of Stirling.
Can you shout, Colin, so that people can...
Colin Gould from the
I'm probably the only deaf person here.
At University of Stirling. I'm fascinating talks. Thank you very much. It's brilliant to see that data being used so well to try and answer some of these really critical questions. A couple of things I'd be interested in exploring, certainly with Bob, is thinking more about what you have from your rotary screw trap in terms of your estimates of your smolt population and annual variation, because that to me would be the big thing that would tell you a lot about your carrying capacity, and this was brought up by the lady at the back. If that's got very little variation from year to year, then it would suggest that your freshwater environment is full, whether it's with stockfish or wild fish will hopefully be looked at when you look at the 200 you have every year. I think that would be amazing to do and really useful for everybody to find out more. But the second thing I wanted to mention there was, do you have an estimate of your rod exploitation rate? Because the sampling of your smolts clearly is very, very carefully controlled and is randomised, and it will be really valuable. But I wonder how that will relate to what you have from your DNA taken from your rod caught fish.
I think well, if I answer first on the screw trap. The problem with a screw trap, and I suspect this is the case for anyone using a screw trap, it only works really well when the river's low. If the river gets too high, fish go past it. We've got it in a narrow section. When the river's right down, it's in quite a narrow section, and a great place to have it, because I can just step off the bank. I don't need to pull it in or have a boat or anything. I can just step off the bank onto it. But if the river gets high, much more the river is passing it. So we wouldn't get the same level of efficiency. The times when I measured the efficiency was when the river was low. So that 16 to 17% that I mentioned was when the river was right down. Also, if your river is running a bit too hard, you'll start getting scale loss on the smolts. And obviously you don't want to be killing smolts. That wouldn't be a very clever thing to do. So it varies depending on how good a run you can have through the season. Now, we tend to put it in as soon as the water gets low enough in April. And it might be in the middle of April, we might not get it until the end of April. So we undoubtedly will miss some early fish going down. We've had seasons where I've been able to run it. I mean, what I have to do if the river rises is just lift the screw. We don't have to move the trap, we can leave that, it'll just sit in the water. But if the screw is still in the water and it gets really high, there's a lot of pressure on that bit of machinery, and on one occasion it disappeared 50 yards down the river with a huge block that it was tied to. It quite amazed me. I just didn't get to it in time to lift it. But there was a couple of seasons when I was able to run it right through, and the numbers we got then were pretty similar. And in any other years that I recorded numbers, we were usually getting, in the years where I could run it for most of the smolt run, we were getting maybe 5,000 to 6,000 smolts going through. And based on the 17%, you're looking at somewhere between 30,000 and 40,000 smolts. Now, interestingly, the management plan that was done for the Carron by the Westeros Fisheries Trust quite a number of years ago, I think it was James Butler that actually did it when he was there. They estimated, based on riverine habitat, a maximum smolt run of 16,500. And that didn't take in the 2 lochs, but in actual fact, I don't believe the 2 lochs produce that many smolts. They're very full of brown trout, and I don't think many young salmon would survive to come out of the 2 lochs. So if you then think, well, if we're estimating that we're maybe getting 30,000 smolts going out, that's an awful lot more than the estimate that was put forward. Now, I suspect that's because a lot of these are the extra fish that we've put in over the years that have increased that number of smolts coming out. But it's not easy. You can have different figures for different years, but then you don't know the differences between the number of times it's been fishing efficiently and not fishing efficiently. So that's quite difficult. Now the other question, what was the other question again, Colin?
Just your rod exploitation.
Now that to me is a very difficult question to answer, because anyone who's a salmon angler or knows people who are salmon anglers knows that not everyone fishes efficiently, not everyone spends all the time fishing all day. Well, ironically, if I was looking to decide on a level of exploitation versus rod catches, to give me an idea of maybe how many fish were in the river, I would look at my catches, because I fish the same way all the time. When I go salmon fishing, I go salmon fishing to catch fish. So I don't necessarily go salmon fishing just to enjoy casting a line. I'm going there for a reason. And I'll be in the river just about every day through the fishing season. When I know there's some fish around, I'll be in the river virtually every day for some part of the day. I will probably be more efficient in an hour's fishing than someone else who maybe didn't know the river, maybe couldn't read the river that well, or wasn't particularly good at reading where to fish for a salmon. And I've seen plenty of anglers in the river that have been fishing in places where I think, you may as well go home, you're wasting your time. So I would take my catches as being a good indication of exploitation rate, because I think it was the same for all salmon rivers. It's not like exploitation rates for net fishing, for example, where we used to have all the nets around the coast. You could work out how long these nets were fishing for. The ones that were using net and cobble, you could work out how many times they're going out, so you could get figures for that. Angling is much more difficult to actually do, especially when you've got non-gillied rivers. A gillied river is slightly different, because they have a gillie to guide them, show them where to fish, what to fish for, and the gillie is probably pretty keen that they catch fish for different reasons. Ian's smiling here. That's not an easy one to answer, and I find when Marine Scotland Science started putting rod effort in to go along with rod catches, my attitude to that was, that's going in just so you've got a figure. That's not really going to tell you that much. But I can go through my catches over the years and over different times, and they vary quite a bit. I mean, going from one year when I caught nothing back in the early days before the stocking program, to a couple of years when I caught 160 in each of those 2 years, the 2 big years we had. Because I was in the river all the time, and people sort of argued that, oh, but it's only Bob that's catching fish, nobody else catches. I say, well, I'm the only one out there. I'm the one who's fishing, and I'm fishing with, and I'll be quite blunt about it, I'm fishing with what I'll consider I can catch a fish with, because that's what I was trying to do, I was trying to catch fish. So my figures would actually be quite telling, if you're looking at the exploitation rate. But to look at your general anglers, where you've got people fishing that are maybe not very experienced, don't know the river, as I say, don't know where to fish, what to use, whatever, because there's nobody there to advise them, then it's quite a difficult one to come to grips with.
Victoria?
Yeah, I'll come in because this is a result I didn't present in the talk, but it's in the report. So one extra analysis I did is something called parent offspring mark recapture, and it's a way of assessing the entire size of a population. So in traditional mark recapture, what you would do is you would go into the river, you would fish out a sample of fish, you'd mark them, and then you'd put them back, and then a week later you'd fish out another sample of fish, and and by looking at the proportion of marked fish in that second sample, you could extrapolate to population size. But instead of doing 2 samples of the same population, what you can do is recapture an individual, in effect, by recapturing its offspring. So what I did is I took all the wild fish from a certain number of years, and then looked at whether we got the offspring of those wild fish in the rod catch in subsequent years. So it's the same thing as I was doing with the broodstock, but with the wild rod caught fish. And so, making a few assumptions, I was able to get an estimate of sampling rate. So it's not exactly the same as rod catch, but it's the rod caught fish that were sampled. And in the first year, I got an estimate of 14%, which is almost exactly the same as what Bob's general estimate is. But the second year, I got an estimate of 4%, and we need to look at why that estimate was such smaller. But I would say, yeah, around about something between 10 and 15% is the exploitation rate in the river.
Thanks. A few more questions. The gentleman with his hand up. Thank you.
Sandy Brender, Chair of the River Trust, convener of the Kilgroves National Park Authority, and like Bob, a fanatical angler. And in the interest of transparency, I holiday on the Saturday of the State, and Bob is very, very kind to give me a personal tour of the hatchery. Fascinating presentation, thank you very much. It does pose the question of next, and some of those questions were posed in the last slide. So Bob, this is a question for you. After, I think the word heroic is the best description of Bob's commitment. After a heroic 30 years, no holidays and not seeing your family, this perhaps time to pause, stop and see what the impact would be with a continuing programme of monitoring.
Personally, I think given the fact that a lot of the issues that we have had and still have, I would almost definitely predict that numbers would drop without the stocking. If you know, going by Victoria's figures on the number of stockfish, around maybe 50% of the fish being caught, and you've got to assume that that's 50% of the fish going back to the river, because there's no reason why you're favouring stockfish as against the wild or the other way around. So if that's the case, then if you have a year, I think 2019 we had over 300 fish caught that year. So we are then assuming that at least 150 of those were stockfish, and your exploitation rate maybe means that you're catching a fifth or a sixth. You know, at 15% would be around a sixth. Then you've maybe got an extra 600 fish going back to the river. And you know, that being the case, then if you take that number of fish out of the system, given the fact that these fish will definitely be going on to spawn, I see no reason why a stockfish going back won't spawn successfully. I've caught kelps in the spring that were identified as stockfish, so they'd obviously gone through the spawning process. I'm convinced that it will drop back. I mean, I've been asked often, you know, now that you've got fish numbers back up, why are you still stocking? And of course the answer is, well, nothing's really changed as far as the problems facing the fish. We've done nothing else other than get some young stock in. So all the issues that they had, and in fact they're probably getting worse because our seal population is getting bigger and bigger and bigger, as it is all around the country. And I must admit, I don't like watching these nature programs where they start enthusing about seals. Anyway, no, I think it would drop back down. I'm getting to a stage in life where I ask myself the question lots of times, there must be something else other than doing this. Unfortunately, I like doing what I'm doing, so I'm still doing it as long as I'm fit to do it. But the answer is, if we stopped, I don't know whether it would start up again. It wouldn't be up to me. It would be up to Sean and the others as to whether they would continue. It would be difficult.
Thanks, well, Victoria, do you want to chip in on how long you'd have for a proper scientific study of leaving the river?
Okay,
how many years you'd have to leave it?
I'll just make one point first. Again, this comes back to the carrying capacity, because we don't know at the moment if if we stop stocking, whether there'll be increased survivorship of wild spawned juveniles and more wild spawned smolts. So that's one of the big questions. We don't know. Well, I mean, that's a good question. How long would we want? I mean, I think we'd want to, like, monitor it for at least one generation, wouldn't we? So that's, at least probably about 5 years, yeah.
It's a long time.
Yeah.
Can we quickly, Victoria, thanks. It's certi had drink frae druth, as they say in Scotland. But we've got a couple more questions. Is it Pritchard?
Pritchard, yes.
Pritchard, can I take your question then? I've got John Gibb at the front. And another question here. I'll come to you in a second. Thanks. If you can be brief, please.
Yeah, Richard Summers, just a very keen angler. I fished the River Carron many times with Sean. So this is a very specific project on the Carron and all fantastic work. Obviously, there's loads of other rivers in Scotland. Every river is unique. I think the Carron is a beautiful, fantastic river to fish, but every river is different. How adaptable is this research to other rivers? And if you're in Marine Scotland forming your policies on hatcheries, how do you take account of every single individual river? For example, West Coast Spatey rivers are clearly very different to the Dee and the Tweed and the Spey. Are Marine Scotland listening to?
Well, I think we know the answer to that question. Just in the interest of a bit of speed, John, can you put in your question, please, and then the panel will attempt to answer them both, and then I'll come to the gentleman here as our last.
John Gibb, I'm the coordinator of the Salmon Scotland Wild Fisheries Fund. I'm also a fishery manager on the West Coast, undertaking quite a lot of stocking, so for various reasons this is obviously fascinating. But can I just quickly preface and say that how delighted the Salmon Scotland board were to underpin this work, because we see it as absolutely vital. And furthermore, I'm really pleased to say that they've committed to funding at least the first part of the Smolt work that Bob's mentioned tonight. So that's going to take place this year, or start this year. So that's very pleasing. But my question is more with my manager's hat on. The report's very interesting for a whole variety of reasons, but it's identified that, stocking works and that the fish come back. But it also identified that for endangered rivers, this might be a very, very useful tool, amongst others. But at the end, you identified that using the captive broodstock might have a long-term issue. Now, my colleague who's just asked the previous question has identified that there's spate rivers up and down the west coast that are in dire straits. If not, the population's already lost. So what would you do if you were managing these rivers? Because you've said that to use a captive broodstock is probably not wise, but these rivers don't have enough wild broodstock in them to take the fish out. So if you're in that scenario, what would be the best advice from a scientific point of view, from a practical point of view, for the many rivers this would apply to?
Yeah, it's really tricky for sure. I will say that we looked at the two different broodstocks. So the US broodstock, I think the parents were only used for a single year, and the Athdale broodstock, the females were kept for multiple years. So the reason that the Athdale broodstock reduced the genetic size of the population was that because you had a lot of females and they were bigger and bigger and they're producing a lot of eggs, but you've got the same mother producing a whole lot of fish, whereas the US broodstock you were using a lot of different mothers. And even though both the males and the females were captive, I know that they were very careful at not crossing closely related fish. So certainly if you had to have a broodstock, having that just use fish once is better than keeping them and using them again and again. And you could potentially, you might even consider releasing them as, reconditioning the kelts and then releasing them back into the river. Yeah.
Good.
Yeah, hi, I'm Ian Bell and I'm from Salmon Scotland and represent Tavish, so he sends his apologies for not being here. Just a comment, I'm really pleased what John said. Yeah, I've been very supportive of this, and I'm really pleased over the years there has been engagement with salmon farming companies on this. I'm not a wild fisheries ecology expert, but I have a question for Bob. You mentioned that there's a high propensity for precocious parr within the system. Have you done any genetic sampling of those, and would there be value in doing that, and would they provide an opportunity for enhancing the genetic diversity of the stocked fish? Bear in mind there may be other knock-on implications of doing it.
Well, I have to say that I've never actually seen a pair of fish spawning in a Carron. I have had a wee look. We have an awful lot of gravel in the lower part of the river from Loch down, and it's not like having just specific spawning sites. There are so many places where the fish could spawn. So I can't answer the question as to how many precocious parr there would be. I'm sort of simply going by what is evident in other rivers, that there are these precocious parr. And I can't believe there wouldn't be in the Carron as well, but there wouldn't be precocious parr there. The whole issue of precocious parr is, and I don't know whether this has been looked at in any sort of scientific way, is it a good thing to have precocious parr fertilizing eggs at all, if that maybe gives rise to more precocious parr? I mean, I've certainly been around precocious parr when I've been rearing fish, and you'll get some that do mature as parr. Would that tend to give rise to more precocious parr if you start breeding with more and more precocious parr? I know it's often seen as a backstop. If you're short of fish, you can always catch up some precocious parr and use them. But I'm not entirely sure. I mean, I don't know if Victoria.
Well, I mean, it partly depends what your aim is. Is your aim to have nice big fish coming back to be caught, or is your aim to save the population from extinction? And those are two different things. And I have to admit, I've really only recently started to appreciate exactly how much precocious parr might be contributing to the next generation. And just because a fish spawns as a precocious parr doesn't mean that all its offspring are going to be precocious parr. And actually there's some really nice work coming out from the work the Atlantic Salmon Trust have been doing on this tracking in the Laxford and other rivers, is that quite often the precocious parr will then mature and go out to sea, and they'll return as grilse or even salmon. So it's really quite a neglected part of the salmon life's history. And potentially, because the survival at sea seems to be going down so much, that is really evolutionary selection for the precocious parr life stage. So I think it's really an important element of keeping the genetic diversity and the wild salmon spawning in the river. And it's true, it's really hard to look at how much of the offspring are contributed by precocious parr. There's been a couple of studies done, and it's surprisingly large amounts. And for example, some of you might know about the restoration of the River Carron in the Tay, and we're getting a lot of wild spawned fish juveniles up there, right in the upper part of the Tay. And I wouldn't be surprised if a lot of that is the offspring of juveniles that were stocked and then spawned up there as precocious parr, rather than going to the sea and coming back again.
Okay, good. I could listen to Victoria and Bob all night. Can I ask you, Sean, just to wind up? I think you're going to mention a couple of our river owners who've passed away.
Very happy to do so. One, thank you all for coming along today. Fantastic. Support Bob, support Victoria and all the fantastic work. We're very grateful to Tavish and John for the support. They came to see us last year and said, yeah, we need to progress. One of the things that going forward we should also be looking at is the grading of rivers on the modelling, because the year we caught 400 fish, there's an assumption that you'll have eggs deposited, but with our red washout issue, we might have been better catching 40 fish and no specks, and we might have ended up with more fry. So basically, the modelling for the grading is not fit for purpose. That's just the way things with the Scottish Government, Edward. Come as no surprise. I've ended up this year writing a couple of letters about my friends who are no longer with us, Mr. Ewan McPherson and Richard Wilson. When we go back in time to when we were at a very low ebb and we weren't catching any fish, I remember going to see Mr. Gordon and Mr. Ewan And we wrote in to tell Marine Scotland that we needed a column for fish farm escapees. And we had to make a decision, and at that time my late father was still alive, and John Braidwood, as to how we were going to approach fish farming in the area and the collapse of the river. And the decision was made that as part of the community, and unusually as we all lived in part of the community, we should embrace fish farming, we should challenge them, we should try and create a Smolt run, and we should try and work with them. And with the help of Bob, we've managed to do that, you know, throughout the last 30 years.